Hi everyone, welcome to the lesson in the fundamentals of chemistry in life sciences. As you are aware, we are talking about building blocks that we encounter that are essential component of our life. In the previous lectures, you have learned about DNA, RNA, you have learned about proteins, you have also learned about nucleotides, amino acids. In this lesson, you will be learning about carbohydrates. Carbohydrates are you can think about carbohydrates as the energy currency. We mostly regard carbohydrate as the major component through which we gain a lot of energy, and we uptake carbohydrate because we need them for energy. But towards the end of this lecture, you'd also understand that carbohydrates are very simple yet very complex, and it not only play a vital role for providing our body the energy, but it also plays an important role in form of glycoprotein, which is essential component for cell-to-cell recognition, interaction, immunity, immune response, and so on. Attachment of these carbohydrates to the proteins determines the protein function, protein recognition, and its regulation within a cellular system. Therefore, this carbohydrate plays a multi-layered role, and it's functionally vital for our existence. Now, a simple question. Can you live without sugar? I think very difficult. You see, whatever we are having every day, starting from soda to cookies to candies to chocolate to ice cream, I'm sure everybody like ice cream. And what they have, it has sugar. And these carbohydrates are included, present in grains, fruits, vegetables, legumes, sugar, meat, you name it. And uptaking enough amount of carbohydrate is essential for us, for all of us to survive. Now, what is the general characteristic of carbohydrate? The term carbohydrate is derived from a French term called hydrate de carbone. And as you can guess, hydrate refers to water molecule. And the reason why it's called carbohydrate, because the molecule is mainly composed of three elements: carbon, hydrogen, and oxygen. And the empirical formula is CH2O, and this block can be present in multiple times, and that defines the nomenclature of this biomolecule. So I'm sure you have learned about carbohydrate in your previous lectures during your high school. But just briefly, carbohydrate is mainly the most important source of energy. Why? Because carbohydrate, when breaks down, it stores the energy in form of ATP, which is what we use for all our functions. They also are the intermediates in the biosynthesis of other basic biochemical entities, fats and proteins. Not only that, carbohydrate are essential component of your nucleotides. Do you all remember purine and pyrimidine that you must have read in your DNA and RNA lectures? what you would remember is the presence of the carbohydrate, the sugar moiety present in these nucleotides. That's one of the important elements. These carbohydrates not only provide the energy or the intermediates of some important biochemical entities, but they also participate in biological material transfer. They also participate for cell-to-cell recognition. They act or they are important factors for growth and for our immunity system. So you can imagine such a simple molecule which has its empirical formula as CH2 block multiple times, CH2O block for multiple number of times, can perform so many diverse functions, from energy to cell recognition to immunity to synthesizing several basic biochemical entities. This just suggests that how these building units are essential part of our survival. These carbohydrates are further classified into monosaccharides, oligosaccharides, and polysaccharides. And I think it's very simple to remember because these are just the Latin terms. Mono represents one, oligo is multiple, polysaccharides is again a unit being repeated several times. And then the examples, as you can see in this case, among oligosaccharides, the most important ones are the disaccharides, normally lactose and sucrose. And one important thing I have forgotten mentioning is that carbohydrate plays a very important role in plant cell. So the fibers that we use, we can find everywhere. These fibers are the building blocks of these fibers are carbohydrate. Isn't it fascinating? Monosaccharides, as the name itself refers to, mono represents one. So these monosaccharides are the simple sugars, and you can think about it as the simplest sugar. And then the example includes, for example, glucose, fructose, galactose, and so on. These are the principal monosaccharides in food, and they are further classified if they have aldose or ketose group, and also the number of carbon atoms they have. Now, as I just mentioned, the aldose or ketose groups are basically a criteria based on which you can define these monosaccharides. So for example, in case of D-glucose, what you see, first carbon atom has the aldehyde group, and that's why this is called the aldose. On the other hand, if you see fructose, the second carbon has the keto group, C double bond oxygen, and that's why this is called as keto group. So like I just mentioned, the monosaccharides are classified based on the number of carbons they have, or rather the unit carbon units, as well as the functional group that are present there. Now in this slide, as you can see here, the monosaccharides that are present, these are the units. So for example, the ribose, deoxyribose, glucose, mannose, galactose, fructose. All of them can be classified as, for example, the function, if they are ketose or aldose. That's number one classification. The number two classification is the number of carbon atoms that are present in them. And the presence of, or rather the number of carbon atoms that are present in monosaccharides, as well as if it has the aldose or ketose functional group, for example, determines the cyclic form that these monosaccharides possess. For example, in case of glucose, the resulting intramolecular hemiacetal 6-carbon ring is called a pyranose because it resembles to pyrene. On the other hand, in case of fructose, for example, what you have is a keto group, and because of which there is a formation of hemiketal, a 5-carbon ring, which appears to be as furan, the chemical organic compound furan. In this slide, what you see is the open chain of the glucose, which then can be formed into two forms, alpha and beta form. And as you can see here, this this group, aldehyde group, that is present as carbon number 1, basically acts as the point at which the OH group of carbon number 5 attaches and forms a heterocyclic, where meaning the oxygen is within the cycle, and the position of OH group determines if this glucopyranose is alpha or beta form. So for example, in case of alpha, you can see clearly the OH group is farther away from the oxygen atom. On the other hand, in case of beta form, the OH group is closer to this oxygen atom. Now, I think you should think about which form is more stable. What do you think? Do you think the alpha glucopyranose is more stable or beta glucopyranose is more stable? Please consider this as a homework. Try and find out which one is more stable and why. Just a bit hint, clearly when oxygen atoms are close enough, they have the lone pair of electrons which repels, and that's why you can imagine as if they are fighting with each other. Most likely that's not stable. Please find out and explore if alpha form or beta form is more stable. Similar to D-glucose, when you look at D-fructose, although the chemical formula is the same, C6H12O6, the major difference is compared to glucose, this is a ketose, so you have C double bond O at carbon number 2, and therefore it forms a 5-membered ring where OH group attaches itself to partially positive carbon atom because of the electron negative oxygen atom connected with it, and this results in the formation of a furan type 5-membered ring, as you are able to see in this case. Now with this, as you can see here, the way these structures are being formed, you will have various forms of these monosaccharides, and that adds up the diversity that exists in case of monosaccharides. So for example, as you see here, the ring structure of fructose can form both 5-membered furanose and 6-membered pyranose rings. In each case, both alpha and beta anomers are possible, and again, as you can see here, alpha and beta, the stability depends upon the orientation, or rather the position of these atoms within the space. Now, glucose are the main source of energy which help us to function. Glycolysis is splitting of glucose. So for example, the 6-carbon of glucose splits into 2 pyruvate or pyruvic acid molecules, and this splitting releases in total 36 ATP molecules. So one glucose molecule through the Krebs cycle, which occurs in mitochondria, eventually results in the formation of 36 ATP molecules. And as you all know, the ATP is the energy currency. Our body preserves the energy in form of ATP and utilizes it whenever it is needed. Therefore, the carbohydrate, for example, glucose or fructose, these molecules are essential building blocks which provides us, which kind of stores the energy from where we get our energy. And that's why their presence in food, or rather uptaking these from food, is how we get the energy for our day-to-day purposes. Now, as you learned, the glycolysis is splitting of the glucose molecule, for example, resulting in release of energy stored in form of 36 ATP from each glucose molecule. Now, in case when you have not taken adequate amount of carbohydrates, your body definitely continuously requires energy, especially, for example, your brain. It certainly requires glucose. Now, in case of inadequate amount of glucose in your body, the protein can start breaking down, and they result in production of glucose. And this process is called gluconeogenesis. Through this gluconeogenesis, your protein starts producing glucose, which then provides you the enough energy for functioning. But what this means is that this is a special situation, and if you are not uptaking adequate amount of carbohydrate, then the protein degradation is going to happen. Therefore, the food should contain adequate amount of carbohydrate to prevent the use of protein for energy. And this role of carbohydrate is called protein sparing action. Now, these individual units, that is, for example, glucose, fructose, and so many examples of monosaccharides we have seen, when they come together and when they are linked via O-glycosidic bond, oligosaccharides are formed. So in a way, you can think of oligosaccharide as the storage form, because whenever we are talking about the using these carbohydrates for the generation or production of energy for our body, these oligosaccharides needs to break down into monosaccharides, and then these monosaccharides are utilized by cell to produce energy. So in the other way around, you can think of oligosaccharide as the storage form of energy or a storage form of monosaccharides. Now we are moving from monosaccharides to oligosaccharides, and as we just discussed, the most common disaccharides includes sucrose, lactose, maltose, and as you could guess, the di refers to two. Therefore, disaccharide means two units of monosaccharides comes together to form disaccharides. And in a way, if you think about it, these are the most generally found means of monosaccharides that we use for our own energy gain. So, for example, sucrose, lactose, maltose. These disaccharides are some of the major source of energy that we intake from our food. Now, maltose consists of two molecules of the glucose, while lactose includes a molecule of glucose and a molecule of lactose, while sucrose includes a molecule of glucose and a molecule of fructose. In general terms, when we are talking about within a biological system attachment of these sugar molecules. You are talking about oligosaccharides, for example. What oligosaccharide means is upon one sugar, one monosaccharide, another monosaccharide comes and are being connected. Now, there in biological system, these attachment of sugar molecules are performed by a set of enzymes called glucosyl transferases. And these enzymes uses, for example, molecules such as UDP glucose. So in this case, as you can see here, for example, in this case, UDP glucose consists of UDP and glucose. And these glucosyl transferases enzyme takes the glucose from UDP glucose and then transfer it to its target molecule. It could be protein, it could be a metabolite, a chemical structure. And therefore, when a sugar molecule has to be transferred, the role of enzyme as well as the role of the carrier of these sugar molecules or these monosaccharides are equally important. The sugar to be added comes from a sugar nucleotide, like as I just mentioned, in this case UDP glucose. The acceptor, for example, in this case designated as X in this slide, can be a simple monosaccharide. It could be even a protein within, so it could be a serine or threonine residue belonging to a protein and so on. So these enzymes uses these sugar nucleotides to transfer the sugar molecule. Here you are seeing structures of sucrose, lactose, and maltose. So as you can see, in case of sucrose, you are getting 2 units of 2 monosaccharides, glucose and a fructose. What's important in this case is the way you read the name. So for example, sucrose would be called alpha D glucopyranosyl beta D fructofrunose. And the reason why this naming is so important, because just by the name you will understand the configuration that exists. So for example, in case of sucrose, the glucose unit is the alpha form, while the fructose unit is the beta form. And the term pyranosyl and furanosyl represents the 6 and 5-membered configuration. If you all recall, fructose can give rise to 5-membered as well as 6-membered ring. And that's why when we say that sucrose is a disaccharide which includes glucose and fructose, the name does give a sense of final structure that we're going to see. Therefore, when you read such full name of a carbohydrate, these names are written in such a way that it reflects what kind of linkage is there. For example, in case of lactose, you can see that the bond is formed via 1 to 4. First carbon of beta galactopyranose is forming a bond with 4th carbon of D-glucopyranose, and such nomenclature is how being practiced in naming these carbohydrates. We all are very well aware of sucrose. That's what we take every day, coffee, tea, whatnot, and they are acquired from different sources, including sugar cane, sugar beets, and so on. Lactose is, as you just saw, this disaccharide forms by joining alpha D glucose with beta D galactose via 1,4 linkage. This lactose is called also the milk sugar, which is the major disaccharide present in the milk. It's sweeter and more soluble than the organic alpha lactose. The beta lactose is sweeter and more soluble than the ordinary alpha lactose. Maltose, the another disaccharide example that you have seen just in a few slides back. Here, 2 glucose molecules are joined via alpha 1,4 linkage. and they are known as the malt sugar formed by partial hydrolysis of starch. And I think most of you are aware that maltose are found quite prominently in beer and malt liquors, and they are used as a natural as a nutrient. For example, in barley you can obtain, and they are also key as a fermentative reagent. Now you have learned about um, monosaccharides, you have learned about oligosaccharides, and the most example we have seen is disaccharides. Now, plant stores glucose mainly in form of a polymer. These polymer means a unit which keep repeating itself, and they are all connected, forming a long chain. This is polymer. So plant stores glucose as amylose or amylopectin, glucose polymers, thus collectively called starch. And so for example, in case of amylose, you have glucose polymer connected via alpha 1,4 linkage. And please remember, 1 to 4 linkage defines carbon number 1 of first glucose forming a linkage with carbon number 4 of second glucose. And this alpha 1,4 linkage is present along the entire length of amylose. So please see here, the naming of these molecules are so scientific. It ensures that you remember, or you can find out what configuration these molecules are, if it's alpha or beta. You can also find out just by seeing the name or noticing the linkage that the polymers are connected from first carbon to fourth carbon through this linkage of oxygen, the linkage that exists resulting in the formation of polysaccharides or oligosaccharides. And this is extremely important because clearly the formation of these bonds, the formation of these biomolecules is required to ensure that each of these chemical structures achieve the most stable energy state. The general characteristics of polysaccharides are they are found naturally in the bound form, and you have several examples. You have polysaccharides, you have glycoproteins. These are the proteins where some of the amino acids, for example serine or threonine, or some of them get linked with these polysaccharides, and then they form a very important signaling molecule. Sometimes they kind of assign a type of marking. Cell-to-cell communication occurs via this whole process of glycoprotein. In fact, these polysaccharides play a major role in determining our immunity. Other than that, as you can see, these carbohydrates plays a very important role in formation of the key building blocks of our life. For example, nucleic acid, which is what is our DNA made from. Now, the branch point in glycogen, for example, in this case, two chains of glucose molecules joined by alpha 1,4 glycosidic bonds are linked by an alpha 1C glycosidic bond to create a branch point, as you can see here in this slide. Such an alpha 1,6 glycosidic bond forms at approximately every 10 glucose units, making glycogen a highly branched molecule. So you can imagine all these polysaccharides. You can think about them in a similar way as you think about a protein. The difference is, in case of protein, you have 20 units. And these amino acids sometimes have different kinds of charges, they have different types of properties, which determines their three-dimensional structure. In case of polysaccharides, the same unit, or for example, the disaccharides, which then repeats in a very ordered fashion, result in formation of your polysaccharides. And in these cases, the formation of the bond, for example, in this case, alpha 1,6 glycosidic bond, these branch points eventually shapes the overall complexity or simplicity that one can observe in a specific polysaccharide. So starch, for example, is one of the most common storage polysaccharides in plant. It composts of about 10 to 30% of alpha amylose and 70 to 90% of amylopectin, depending on the source. And please understand this fact that the composition of amylose and amylopectin would eventually determine the kind of, the type of, the strength of starch. And this composition is what also creates differences. It creates a kind of different properties between plant to plant. So that is what it means when we talk about depending on the sources. And then, of course, I'm sure all of you are aware of the common sources of starch are, for example, grain, potatoes, peas, beans, wheat, and so on. Glycogen is also known as animal starch, stored in muscles and livers. And as you remember, in previous slides, we mentioned about glycogen that they do not stay for longer time, and that's why we need to keep uptaking carbohydrate to fulfill our energy need. These glycogens are stored in muscles and liver, for example, and most glucose units in glycogens are linked by alpha 1 to 4 glycosidic bond. And you also have to understand that the formation of these linkage bonds sometimes have parallel bonding that causes a very complex branched structure of these polysaccharides. The storing glucose as glycogen, I mean, the glycogen, you can think about glycogen as the storage form of glucose. In fact, 1/3 of total glycogen is stored in liver and 2/3 in muscles. Now, as we have discussed, and as I was mentioning when I was talking about the monosaccharides and disaccharides, the energy is released when monosaccharides are, for example, acted upon, say, through glycolysis process, for example, in mitochondria, resulting in 36 ATP from one sugar molecule, one glucose, for example. Now, these polysaccharides are the storage form of these monosaccharides, and that means when you need energy, these polysaccharides are degraded, results in breaking down and getting the individual monosaccharides, which is then utilized for the energy creation. Therefore, for example, when blood glucose falls, the liver cell break down glycogen into single molecule of glucose, which is then made available to supply energy to our central nervous systems, our brain, and our organs. And during exercises, clearly the muscle cells themselves uses up the glycogen they store, and that basically is what, we are, when we are performing some sports activity or some exercise, the glycogen is then breaks down to glucose to provide us the energy. This is, for example, one representation of starch and glycogen, where the linkage is alpha 1 to 4 linkages, and these units are going to be present multiple times. Fibers, on the other hand, is found in food derived from plants. As you are aware of, there are several plant-derived food which are rich in fiber. These include polysaccharides such as cellulose, hemicellulose, pectins, gums, mucilages, and uh, again, it depends. I mean, these polysaccharides are these polysaccharides consists of monosaccharides forming a linkage, and these polysaccharides can be considered as the storage form of monosaccharides. But you see, if these polysaccharides cannot be broken, it cannot be used to get individual monosaccharides, then it's of no use. So for example, fibers, these are not source of energy for human, because our digestive enzymes cannot break down these fibers. But then there are some so for example, cow, and there are several animals where they have the digestive system that can break down these fibers, and that therefore these monosaccharides can be utilized for the energy creation. Now please notice, in this case, this is the cellulose where you have beta-1,4 linkages, and in this case you can think about it as, for example, here the glycosidic bonds determines the polysaccharide structure. So clearly the beta-1,4 linkages favor straight chain, which is what you can see here, which is optimal for structural purposes. But at the same time, the alpha-1,4 linkages favor the bent structure, which are more suitable for storage. So the different the amount of these linkages that exists within a given polysaccharide determines its properties. Meaning, if this polysaccharide, for example, there could be a polysaccharide where it is rich, dominated by beta-1,4 linkages. That means most likely that specific polysaccharide is optimal for structural purposes. On the other hand, alpha-1,4 linkages favors a bent structure, which is then more suitable for the storage of energy or storage of glucose and so on. So we have actually talked quite a lot about these carbohydrates as a source of energy, but carbohydrates play another important role, which is by being attached to the protein, they forms the glycoprotein. In fact, the glycoproteins are the largest component by weight, and they play a very important role, including as the membrane proteins. The important thing with these glycoproteins are the combination of the sugar molecules that are attached with the protein determines, or it works as a signature or identifier, and therefore they play a very, very important role in several times determining post-translational modification. It also determines cell-to-cell interaction and many functions that the protein plays. These attachment of sugar molecules are very critical. Carbohydrates are so the glycosidic bond is what then connects proteins with the carbohydrates. A glycosidic bond links a carbohydrate to the side chain. For example, in this case, a glycosidic bond links a carbohydrate to the side chain of asparagine, or to the side chain of, say, serine or threonine. The glycosidic bonds are shown in red, and as you can see here, these linkages is how, so what's actually happening is within a protein you need to attach these oligosaccharides, or you can say and sometimes polysaccharides, but mostly oligosaccharides, to these protein structures, and this is how the protein can be identified or is identified within a cell. So in this case, you can see this is N-linked oligosaccharide. In this example, what you are seeing is this is the asparagine, where a pentasaccharide core, the pentasaccharide core is actually shaded gray color in this case. Pentasaccharide is basically have five, so it's a core of five sugar molecules of pentasaccharide. So this pentasaccharide core is common to all N-linked oligosaccharides, and these serves as the foundation for a wide variety of N-linked oligosaccharides. Here, as you can see, some of these examples. So for example, this A is high mannose type. So as you can see here, this pentasaccharide core has is dominated by the presence of mannose. For example, the B in this case is the complex type. So as you can see here, the combination, the type of sugar molecule, the core that exists and that connects these proteins, like for example in this case the amino acid which is used for the linkage of these oligosaccharide is key, which determines the overall function and property of that glycoprotein. Now these, for example, this illustration represents oligosaccharides. As you can see here, these colorful, again here, this is a pentacore, in this case connected with asparagine. You can also see some of these oligosaccharides are linked with serine in this case. So what you see here, this example represents the oligosaccharides being attached to erythropoietin. Erythropoietin has oligosaccharides linked to 3 asparagine residues, as you can see here, ASN38, ASN83, and ASN24, and 1 serine residue, which is serine 126. The structure shown in this slide are approximately to the scale. The carbohydrate structure represented in the amino acid residues are depicted symbolically by employing a scheme in this case. And as you can see here, that the combination of glucose that are present would determine, and actually it would change even the function, role, position, placement within the cell. And that's why glycoprotein plays a very important regulatory role in our body, for our body. And these oligosaccharides, or the binding of these carbohydrate to the protein, serves as recognition site as well. So for example, lectins are specific carbohydrate binding proteins. Now we are all in this era of COVID, and I think all of you are very well aware of the spiked structure of COVID-19. And what you know already is the virus code. The protein is linked with several of these glycoproteins, and these glycoproteins serves as a recognition site where our cell do not want the virus to enter, and viruses adapt, modify these glycoproteins in such a way so that they will be able to enter. So some viruses, for example, gain entry into cell by first binding to the carbohydrates on the cell surfaces. For example, influenza virus, they bind to the oligosaccharides composed of salicylic acid and galactose. Similarly, the viral protein hemagglutinin is responsible for carbohydrate binding. So actually, our body the immunity of our body uses these glycoproteins or these signatures as a way to define if something is foreigner or if that belongs to that cell or that system. But the successful viruses are those which can then break that recognition system. Um, so for example, in this case, you can see in this illustration, you have a host cell membrane where the lipid bilayer you observe in this cell membrane, what you have is the glycoprotein. Now these proteins are linked with several of the sugar moieties, which then interacts with the which then interacts with the attached protein within the virus coat. So influenza viruses targets cell by first binding to the salicylic acid residue, the purple diamond that you see located at the termini of oligosaccharides present on cell surface glycoprotein and glycolipids. These carbohydrates are bound by hemagglutinin, the black interaction ovals, as you can see here, one of the major proteins expressed on the surface of the viruses. The other major viral surface protein, neuraminidase, is an enzyme that cleaves oligosaccharide chain to release the viral particle at a later stage of the viral life cycle. So as you can see here, this is more of like a cat and mouse game where these glycoproteins are they serve as the cell recognition system. At the same time, the viruses attack these glycoprotein, and if they can invade, they will be able to enter into the body and cause the disease. And like one of the approach our body takes is they uses these oligosaccharides kind of a pattern matching, and this pattern matching when fails is when our body regards something as a foreign and completely rejects. So one of the major issue we are, I think many of you are aware, is something called organ transplant, which often gets rejected because, for example, if we are harvesting or acquiring organ from some other individual and then trying to implant in another individual, the the dissimilarity of these glycoprotein uh, results in identifying the organs as a foreign element, and that's why our body or the patient's body rather would not accept these organs as its own, and that results in the rejection of that organ. Now um, think about it as an immune response. Our body needs to understand what belongs to it and what is foreign. And that's why I have mentioned that the glycoproteins are important factors. These are the important determining factors which assigns identity of certain protein, which assigns the communication between them, transport sometimes, immune response, and so on. With this, we come to the end of this lecture. I would like to summarize very briefly, very quickly. We started this lecture on carbohydrate, where we mainly focused on the energy and the structural aspect of carbohydrate. I would say more towards the functionality or functional application of carbohydrates. We learned about different kinds of carbohydrate, its function towards the energy as an energy source. We learned about monosaccharides, disaccharides. We learned about oligosaccharides, polysaccharides. We also learned that monosaccharides are classified based on the number of carbons they have. They are classified based on the aldose or ketose group, and then that they are having linear as well as cyclic forms. We then learned about that what role monosaccharide plays, for example, releasing the ATP molecules, which is then therefore needed for our energy source. We also learned that carbohydrates, they are important for synthesis of several important biomolecules. These includes nucleotides, as you all are aware of the fact that these nucleotides are needed for our formation of DNA, which is the genetic information is contained. That's how a living being is defined. We also learned about how monosaccharides forms disaccharides. We learned about sucrose, maltose, and we learned the different combinations that are there. We next then followed it up with the oligosaccharides, polysaccharides, their function, the different combination that exists. Towards the end, we very briefly talked about glycoprotein. I wish I could have explored more detail about glycoprotein, but glycoprotein by itself is such a big topic. I would recommend, if some of you are interested, please explore this topic in more detail, because these glycoproteins is a very important regulatory aspect of a cell. And these, you can imagine the combination and the order of these oligosaccharides basically serve as you can think about it as an identity or fingerprint, and that determines their function. We also talked about, for example, the reason why the organ transplant sometimes fail, because of the recognition fails within a system. Now, I would recommend you all to check carbohydrate chapter in La Ninja. It's explained in much detail, and explanation is very, very easy. Like I said, I would like you all to request, if possible, please focus on the structural aspect, meaning, for example, why alpha is stable sometimes or beta is stable, the linkage that is present, why some carbohydrates are more towards the structural support type, whilst other carbohydrate or other polysaccharides are more the storage of energy types, and so on. These are the fundamentals, and if you could explore it in more detail, you would be more knowledgeable while applying it in your research. Lastly, normally I would have loved to give and explain this topic in presence of all of you, but this is video on demand. So in case some of you need more explanation or you have many questions and so on, I would recommend, if there are 10 or more people who need more explanation or they need more detail, what we can do is I can try and arrange a Zoom discussion maybe for 30 minutes or 1 hour, where I would try to explain as many questions as you guys have and provide you a more detailed description on this topic. With this, I would say thank you so much. I wish you all the best and happy learning. Thank you.